I now invite Mr. Alexander Masny. Mr. Masny is a biochemist with a Master's of Science in Life Science Informatics, and he's a representative of the Scientific Advisory and Research Council of the ALLATRA Global Research Center. Mr. Masny, the floor is yours.
Thank you. Thank you so much. It's a great honor to speak here today on this topic. And we'll have a look about what is known of the actions of nanoplastic particles and their electrostatic charge properties inside a living cell. As Dr. Ann already mentioned, there is much evidence today of potential accumulation of nanoplastics in the brain. But what is happening there at the cellular level within neurons? To better illustrate this, we'll now watch a short animation which schematically will demonstrate how electric charge of nanoplastic particles could influence living cells. So let's have a look at neuron signaling. Each neuron works like a tiny battery. It keeps a small difference in electrical potential between the inside and the outside of a cell. This allows it to send signals at the right time. Charged nanoplastic particles in it, in their turn, may disturb this balance. And the neuron can then behave irregularly, activating too early or failing to send a signal at the right time. If this happens repeatedly, communication between neurons becomes less precise, and the function of larger neural networks can be affected. Every neuron is surrounded by a membrane that contains small gates, so-called ion channels. When the neuron needs to transmit a signal, some of these channels open, allowing these charged particles, ions, to move rapidly into or out of the cell. This movement changes the electrical potential across the membrane and generates a brief electrical impulse. The impulse travels along the neuron and passes the information to the other cells, other neuron. Plastic particles may interfere with the normal function of ion channels. Because they can carry an electrical charge, they may interact with these gates, with the channels, and slightly alter their structure. As a result, the gates may not open when they should, or they may stay longer open when it's necessary. This disrupts the precise electrical signaling required for proper communication within the nervous system. One important finding about micro and nanoplastics is their potential to affect mitochondria inside in the cell. Mitochondria are often described as a cell's energy center. They produce the energy the cell needs to function, but they also help regulate metabolism, cell growth, and division responsive to oxidative stress, inflammation, and even program cell death, apoptosis. Because of the very small size and electric charge, nanoplastic particles can enter cells and reach even mitochondria. There, they may interfere with a normal mitochondrial function. When mitochondria stop working properly, it means the cell can no longer efficiently produce energy or carry out necessary functions, essential processes. Studies from 2025 show that plastic helps bacteria develop resistance to antibiotics, driving a crisis that medicine is increasingly unable to control. Why? Because plastic is the perfect platform for bacterial growth. When bacteria comes into contact with microplastic particles, they can activate a metabolic process related to electron transfer. In simple terms, we can imagine that plastic particles may exchange electrons with bacteria and act as an additional source of energy, sort of power bank. This mechanism is known as an extracellular electron transfer. And by gaining extra energy in this way, bacteria may grow more efficiently, divide more rapidly, and form dense, multi-layered biofilms. Such biofilms can reduce the penetration of antibiotics and make infections more difficult to treat. The range of diseases associated with the destructive impact of micro nanoplastics in the human body is extensive. And it's important to say that when we do not yet have done longitudinal human studies directly linking micro nanoplastics burden to specific clinical outcomes, however, we can say that the presence of micro nanoplastics in organs is confirmed. And in vitro mechanistic modeling and animal study shows that the harmful mechanisms of cell damage caused by micro nanoplastics are biologically plausible. I would like to briefly highlight several categories of disorders, some of which are currently demonstrating particularly rapid growth and affecting a substantial proportion of the population. Neuronal damage is associated with the development of severe, often irreversible conditions, including epilepsy, stroke, Parkinson's disease, Alzheimer's disease, and other neurodegenerative disorders. Having previously worked and a research institute in Fraunhofer on this topic on neurodegenerative diseases, including Parkinson's disease and Alzheimer, I'm particularly aware of really this complexity and societal impact of these conditions. So this topic is a really special issue of scientific relevance to me. And next, we'll see that the work of researchers of the University Medical Center of Utrecht have demonstrated that immune cells exposed to nanoplastics die approximately 3 times faster than unexposed cells. And there are many more studies there. The impact, for example, have been associated with the development and exacerbation of asthma, progression of chronic obstruction of pulmonary disease, pulmonary fibrosis, the replacement of healthy lung tissue with SCAR1, and increased risk of malignant tumors of the respiratory tract. Micro and nanoparticles have been identified within atherosclerotic plaques, dense deposits in the arterial walls that narrow blood vessels and may obstruct natural blood flow. Their presence is associated with increased risk of myocardial infarction and stroke. Studies indicate that when microplastics are detected in plaques of the carotid arteries, the risk of major adverse cardiovascular events and mortality increased by approximately 4 and 5 times. A key mechanism may involve mitochondrial dysfunction induced by nanoplastic exposure. The accumulation of mutations in mitochondrial DNA combined with disruption of programmed cell death pathways reduce the body's ability to eliminate damaged cells and increase the likelihood of malignant transformation, cancer. Experimental studies in laboratory animals have shown that micronanoplastics can accumulate in the testes, induce testosterone deficiency, and impair spermatogenesis. In animals subjected to chronic microplastic exposure, erectile dysfunction was disrupted within as little as 1 month. Equally concerning findings relate to female fertility. A 2025 study identified that microplastic particles were found in ovarian follicular fluid of 14 out of 18 women examined, with an average concentration exceeding 2,000 particles per milliliter. These data indicate that micro and nanoplastics interfere with the earliest stages of life, even prior to conception. In fact, we observe an overall decline of cognitive capacity. Age-related chronic diseases are increasing diagnosed in younger populations. Last but not least, reproductive capacity is being increasingly compromised. The ability to conceive and give birth to healthy children is constantly decreasing. In summary, micro and nanoplastics can no longer be considered merely a matter of environmental pollution. Substantial evidence indicates that it constitutes interference with the fundamental mechanism of human life. What is therefore at risk is not only our present condition, but the well-being of future generations. This topic is now being continued by Professor Ragusa, who will address the impact of children's health. John will introduce Dr. Ragusa, a true pioneer in this field, and Dr. Ragusa will speak on the concept of cyber children, the presence of micro nanoplastics in placenta, and their impact on reproductive function.